Hello everyone, and thank you for joining us for the Cell Press webinar on Organoids and Beyond, 3D Tissue in a Dish. I'm Shri Narasimhan, Scientific Editor at Cell, and I'm here today with Marie Bao, Editor of Developmental Cell, and we'll be your moderators for today's webinar. Over the last decade, stem cell biology has grown from 4 factors that can take a fully differentiated cell back to a pluripotent state to encompass work in many new paradigm-shifting directions. Advances in stem cell research have provided us with unprecedented platforms to model fundamental aspects of development and disease across a range of tissues and organ systems. A central pillar of these advancements is the advent of organoid technologies. Stem cells have the remarkable ability to self-organize in 3-dimensional space into organ-like structures. By harnessing this property, researchers have been able to create organoids from a myriad of tissue and organ sources, both healthy and pathological. These organoids have shed light on a variety of topics, such as the fundamentals of liver development and regeneration, or the mechanistic differences in colorectal cancer progression between individual patients. Organoid models have been powerful tools that allow us to move closer towards recapitulating the complexity of tissues and organ systems.
We're excited to have with us today 2 outstanding experts in the field, Madeline Lancaster, Group Leader of the MRC Laboratory for Molecular Biology, and Omer Yilmaz, Assistant Professor of Biology at MIT. Madeline will discuss how organoids can be used to study a complex organ like the brain, and Omer will tell us about how intestinal organoids can be used to provide unique insights into the pathophysiological processes such as obesity and cancer. These and other work in the field provide exciting implications for regenerative and personalized medicine. They also highlight the road ahead for this technology and the challenges that need to be addressed to accurately model and facilitate the study of complex biological processes. Beyond the webinar, if you're interested in learning more about organoids, Shri and I are also hosting a 1-day symposium called The Organoid Revolution, Organs in a Dish, in Amsterdam on Tuesday, September 27th of this year. We hope that you'll be able to join us if you're in the neighborhood. You can find out more information about this meeting, this lab links, at cell.com/lablinks.
Now, before we get started with the talks, we have a few logistical points. Each of our speakers will give an approximately 15-minute presentation. Following the 2 talks, we will open the floor for a Q&A session that covers both presentations. If you have a question for either speaker during or after the talks, just click on the question tab at the top of the display and type it in. You can enter a question at any time. There's no need to wait till the end. We will try our best to pass on as many of your questions as possible to the speakers during this Q&A session. Also, if you have any technical questions, click on the live event tech support button. We're grateful to our speakers for making time today to join us and our sponsor, Bio-Techne, for supporting this webinar. And now I will pass the ball to our first speaker, Madeline Lancaster. Madeline, thank you for joining us today.
Thank you. It's really a pleasure to be able to speak to you today, and also to be able to give a talk in such an interesting and new kind of forum for me. So I will be talking about neural organoids, or brain organoids, and a variety of different approaches that have recently been developed and their use for modeling human brain development in the dish. So I think most would agree that the human brain is one of the most complex organs in the animal kingdom. And this complexity arises from the fact that it's both quite enlarged compared with other animals, which I hope you can see on this slide. You can see how much larger the human brain is than a mouse brain, but also its density, because even when you compare it with animals that have even larger brains like elephants, the human cortex still has even more neurons than the elephant cortex. So the human brain is both large and very densely populated with neurons. And yet there's still a lot we don't understand about human brain development and how this neuronal complexity arises, particularly because most of what we know about mammalian brain development comes from studies in mice, which, as you can see, don't really model many of the complex aspects of the human brain. And so before I get into some of the features that we're interested in and the model systems that have recently been developed, I just need to give a very brief overview of brain development and some of the interesting features that are thought to be different in humans compared with, for example, mice. So on the left of this slide, you can see a progression of human brain development. And all vertebrates, the brains of all vertebrates start out as actually an epithelial tissue called the neural tube. which you can see at the top there at the 25-day time point. And this neural tube then expands at varying rates on the anterior to posterior axis. And you can see that particularly the most anterior part in humans, which will then give rise to the forebrain and the cortex, expands much more than the more posterior region where, for example, the spinal cord arises. And because this tissue is actually epithelial, the cells that give rise to it, the neural stem cells, are actually epithelial cells. And so in the middle panel there is an image of some transfected neural stem cells of a mouse brain, where you can see this epithelial nature. They have these long processes that extend to the bottom surface of the ventricle and all the way to the outer surface of the brain. And these cells then also divide at the ventricular surface, which you can see in this picture as well. And so on the right is just kind of a schematic showing you how neural stem cells generate the various neural types. They can divide symmetrically to generate more neural stem cells, which we also call radial glia, but they can also divide asymmetrically to give rise to another radial glial neural stem cell in order to self-renew, but also a second daughter cell that would be more differentiated. This could be a neuron directly, and the neuron then uses the basal process and migrates to the outer surface of the brain to generate the cortical plate. The cortical plate will then expand and generate the gray matter of the entire brain. But neural stem cells can also generate intermediate populations, such as intermediate progenitors or outer radial glia. And these cells can divide further to generate even more neurons and therefore expand the number of neurons that are generated. And so one possible mechanism that could lead to a greater number of neurons being produced in the human brain would be through an expansion of these intermediate populations. And in fact, that is suggested to be the case because humans have much expanded numbers of these cells. And in fact, they're so expanded that they form a separate layer called the outer subventricular zone, which is completely absent in mice. So as you might imagine, it's a bit difficult to use mice then, which have been sort of the go-to model system for decades to study these types of questions, these kind of more human-specific types of questions, and also to try to understand features of human disease. And so for quite a while now, people have started looking into in vitro approaches, and more recently even 3D approaches. And so this slide is meant to just kind of try to give you a very basic overview of some of the important methods that have been developed, and that the newest methods are also based upon, of course. So as early as 1989, the first neural stem cells were cultured. And then in the 2000s, so in 2001, the first neural rosettes were generated. And these were really quite an exciting development, because these actually displayed the epithelial characteristics of neural stem cells. And because they also showed this epithelial nature, they could also replicate the lineage of a neural stem cell that it would normally have in vivo. And then in 2008, the group of Yoshiki Sasai made quite a jump with the generation of this method called SFEBQ, which is kind of a 2.5D method where they generate these aggregates and then plate them down and get these really quite beautiful, very expanded rosette-like structures that formed even zones, progenitor zones like ventricular zone and then the outer neural regions. And then again, the group of Sasai made the jump into an entirely 3D, the first entirely 3D neural tissue, and that was the optic cup organoids generated in 2011. More recently, in 2013, myself, while I was in the lab of Jürgen Knoblich, we generated this method, which we termed cerebral organoids, but we also call them whole brain organoids, because this approach leads to 3D tissues with a variety of different brain regions, which I'll discuss a bit further. And now, within the past couple of years, several other methods have been established, and here's one example. For example, these cortical spheroids developed by Sergiu Pasca, where they're able to generate very clean cortical tissues in 3D. And so just to give a bit of an overview of a couple of these methods to give you an idea about how these various methods are similar, but also how they are different from one another. This just shows three methods. I'm focusing here on approaches that lead to the formation of forebrain or cortical tissue, although there are a number of methods, again, particularly from the group of Yoshiki Sasai, where they've been able to generate other brain regions such as hippocampus or cerebellum. But here I'm just focusing on the cerebral organoids, the forebrain organoids from Kadoshima et al., who part of a Sasai group, and cortical spheroids. And so what you can hopefully see is that all of these approaches start with human pluripotent stem cells, such as ES cells, and then go through and then immediately are used to form aggregates, which develop entirely in 3D and eventually lead to these organoids. So in the cerebral organoid approach, there are no growth factors, which is one very important feature, and therefore these tissues actually completely spontaneously form the various brain regions. And they do that by first going through this embryoid body stage, which is a spontaneous differentiation into the various germ layer identities, and then through the use of a neural induction media, which is a very minimal media and kind of prevents the growth of other germ layers, these tissues primarily form neuroectoderm, which is the precursor to that neuroepithelium. And then when we transfer these tissues into a droplet of matrigel, and matrigel I'm sure you'll also hear more about from Omer, is of course one of the key components in many of these organoid approaches. It's a gel of extracellular matrix, and it allows these ectodermal tissues to form buds of neuroepithelium, which then expand quite extensively and form a variety of different brain regions once the tissues are placed in some type of agitation, such as a spinning bioreactor. The forebrain organoids starts out in a very similar way, but there's the addition of a couple of inhibitors, a Wnt inhibitor and a SMAD inhibitor, which actually drive the tissue more towards a neural identity and even affect its patterning, such that these tissues end up being quite pure forebrain identity. And then this approach uses a higher oxygen in order to overcome the issue that we sort of overcome with our agitation in order to allow for better oxygen exchange. The cortical spheroid approach similarly uses inhibitors of signaling in order to drive identity. In this case, they're using dual SMAD inhibition, as well as then later the addition of FGF and EGF and other growth factors even later, which then leads to very pure neural tissue and pure, particularly cortical tissue. And so if you look at these different organoid approaches and what types of tissues they generate, so because the cerebral or brain organoids don't use any growth factors or small molecule inhibitors, these tissues end up becoming quite heterogeneous, where you end up with a number of different brain regions, such as retina here, which you can see in this pigmented region, as well as in the section below it, but also choroid plexus here, marked by TTR, which generates the cerebral spinal fluid. We also do see large forebrain regions, which include both dorsal, which would be the cortical tissue, as well as ventral, and we even see midbrain and hindbrain. The forebrain organoid approach, as you might imagine, generates fairly pure forebrain, which is marked here by the marker FOXG1, whereas the cortical spheroid approach generates more specifically the dorsal forebrain or the cortex, and you can see that here marked by the TBR1 marker here, which shows you how these are organized into many rosettes within a larger aggregate. So because of the nature of these different methods, what we find is that there's kind of this balance between the complexity and the homogeneity. So although the whole brain organoids are quite nice in terms of their complexity and the fact that we can see all these different brain regions, because it is spontaneous, it's very heterogeneous, and each organoid may generate a different complement of brain regions. Whereas as you go towards more less complex tissues, let's say, you get more homogeneity. So the most homogeneous system would be a neural stem cell line, where you have a homogeneous population of single cell type. But you can get something in between, such as 2D neural rosettes or the cortical spheroids or forebrain organoids, where you start to get increasing complexity, but with it increasing heterogeneity. So it's something to consider when one is thinking about these methods and potentially implementing them. But not only is there this effect with, or this balance between heterogeneity and complexity, but even when you focus in on specific brain regions, you see differences. So typically with our whole brain organoids, we kind of deal with this heterogeneity by just looking at those regions that we're interested in, and we are particularly usually interested in cortical tissue. So here I'm just showing you some comparisons of these. First of all, you can see that overall the size of the organoids is very different for these different methods, which you might imagine. So you can see the cortical spheroids are quite a bit smaller than the forebrain organoids, which are quite a bit smaller than cerebral organoids. And that probably comes from that complexity issue. But not only that, when you focus in on specific regions, so in the bottom panel is a trace where we've done tracings of cortex, just the cortical tissue, from data from particular papers, which you can find in this review, and looked at the examples of the largest cortical tissue to try to see what's the largest cortical tissue and what's the most complexity that these can generate. And you can see that as you go from neural rosettes all the way to whole brain organoids, you see an increased size of cortical tissue. And I think this might be because there's this balance between complexity and homogeneity, but also if you think about it in vivo, these various brain regions don't develop in isolation, and it may be that there's quite a bit of communication between them. But overall, all of these methods are able to recapitulate a number of really interesting features of brain development. So I mentioned already the epithelial nature of these neural stem cells, and here are just a couple of examples in both the cerebral organoids and the forebrain organoids. You can see that these neural stem cells also exhibit that morphology. They have this long basal process. And not only that, they undergo a stereotypical behavior of these radioglial neural stem cells, which is called interkinetic nuclear migration, where they actually move their nucleus up and down, timed with the cell cycle, so that they can undergo mitosis at the ventricular surface. And I think you can see that here in this video from a radioglial neural stem cell moving its nucleus along, but they've also observed this in the forebrain organoids as well. And then when it comes to the lineage of these cells, so the radioglial neural stem cells are able to generate intermediate populations. For example, you see intermediate progenitors here marked by TBR2 in the forebrain organoids. We've also been able to identify outer radioglia or basal radioglia, which are marked here by SOX2 and are located in a region separate from the ventricular zone, which is reminiscent of the outer subventricular zone. They're also able to generate, of course, neurons. Here you can see in the bottom left an example from cerebral organoids where the neurons are marked by TBR1 and MAP2. And in the bottom right, an example from cortical spheroids where the neurons are marked by nu-N. In terms of neuronal organization, actually these tissues are able to exhibit quite remarkable, at least immature type of neuronal layering. So in vivo, the cortical plate will expand quite extensively and form 6 layers. Now, although that hasn't been observed yet in any of these organoid approaches, what has been observed is a separation of the more deep layers and the more superficial layers. So in the cerebral organoids, we've seen this with the marker CTIP2, which marks the deep layer neurons, and the marker SAPP2, which marks more superficial layer neurons. In forebrain organoids, they've also seen a similar trend where here brain 2 is a marker of more superficial neurons. And as well in the cortical spheroids, where again you have these deep layer neurons marked by TBR1, more on the inner region of the organoid, and brain 2 marking the superficial layer neurons, more on the outer region. Finally, in terms of neuronal maturation, so a number of studies have started looking at this. So we've done some fairly simple, just calcium staining, where we've put on this fluo4 dye, and we can see this a nice spontaneous firing of neurons here, which you can hopefully see in this video. And this seems to be dependent on neural activity, as we can activate it with the neurotransmitter glutamate, which you can see on the right, but we can also inhibit it with the blocker tetrodotoxin. For the cortical spheroids, they've taken this further and actually done electrophysiology, which you can see on the bottom left, and shown that the neurons that are produced are electrophysiologically active, and they've also looked at markers of synapses. And finally, although it hasn't been examined yet in brain organoids or in the forebrain organoids, the cortical spheroids have been looked at for non-neural brain types. So, for example, astrocytes here marked by GFAP, which you can see have a very nice morphology, and these cells are generated quite late in the protocol. So you can see a peak at about day 181, which also fits with the in vivo timing, as these cells in vivo are generated after towards the end of neurogenesis. And so with that, I'll just finish with a couple of take-home messages. I hope I've been able to show you some of the features of these various methods, but also come across with the idea that there is this important balance between homogeneity and complexity, and it's particularly important when considering which type of method you might want to use. And then I think all of these methods, so very nicely, are able to recapitulate cortical neurogenesis and the behavior of the radioglial neural stem cells. And there is some rudimentary organization of the neurons. They are mature. They seem to form synapses. There are also non-neural cell types. And I think now we have to think about where we go from here. So, for example, taking these various methods to later stages of development, such as getting even better neuronal organization, ideally even 6 layers, and even network formation. I think these types of issues will require probably overcoming an even greater issue that many in the whole organoid field are tackling, which is the fact that there is no vascularization. And so that's something I think that we will need to start thinking about. And particularly for the whole brain organoids or the cerebral organoids, I think ideally one could get both the complexity and the homogeneity. If you think about it, the human brain develops complexity reliably every time. So there should be a way to generate these types of complex tissues in a reliable fashion, and I think that's something that we'd like to do in the future. And so with that, I'd just like to say thank you. It's a really great opportunity to talk at this, and to say thank you to the members of my laboratory here in Cambridge, as well as particularly Jürgen Knoblich, where I did most of the work establishing the cerebral organoid method, and members of his lab, as well as our organoid unit, the MRC, and the NC3Rs for funding. And thank you.
Thank you, Madeline. That was great. Now we're going to move on to Omar Yilmaz. Omar, thanks so much for being with us.
Hi, great to be here. So today I'm going to talk about work that my lab has been doing, trying to utilize intestinal organoids to understand how, for example, a model of obesity impacts the early steps of intestinal tumorigenesis. So over the past 2 to 3 decades, we've really begun to understand the genetic steps necessary from going from normal intestinal mucosa to invasive cancer, where we now understand that there's sequential acquisition of oncogenic mutations, such as loss of tumor suppressor genes and activation or acquisition of oncogenes. But a question that my lab is really interested in is, how does diet impact these various steps of intestinal tumor formation? And we're really interested in understanding how diet is changing the biology of cancer at these various points of development. And today I'm going to talk about a story that we've recently published on, trying to understand how a pro-obesity high-fat diet changes the normal intestinal mucosa, and how these changes contribute to early intestinal dysplasia. So why study obesity? Well, obesity is a diet-modifiable risk factor for cancer, and it's believed that after smoking, diet is the second leading modifiable cause for many different types of cancer. And this has been really understood in a lot of epidemiologic data over the past couple decades, where, for example, in a tissue we study in the lab, the intestine, both men and women who have a body mass index of greater than 30 have a modest but significant increase in the incidence of intestinal tumors. So why is that? So, as I mentioned, our model system for addressing how a pro-obesity diet impacts early intestinal tumor formation is the mammalian intestine, and we model this using mice. And, thanks to pioneering work by Hans Klevers and colleagues, we know that LGR5 identifies most, if not all, of the intestinal stem cells of the mammalian intestine, and using a genetic approach where one knocks in GFP together with a Cre recombinase into the LGR5 locus, one is able to prospectively identify these LGR5 positive stem cells as being GFP high. These GFP high stem cells reside at the bottom of intestinal crypts, shown here in green, and these GFP high stem cells give rise to GFP low transit amplifying cells, which are non-stem cell progenitor cells, which go on to give rise to the cellular diversity of the intestinal mucosa, both in the small and large intestine. Moreover, at the bottom of these intestinal crypts, these LGR5 positive stem cells reside next to an epithelial niche. And in the small intestine, these are Paneth cells, and in the colon, there are Paneth-like deep secretory cells, which are believed to be really important in modulating the biology of these LGR5 positive stem cells and enabling them to not only self-renew and give rise to other stem cells, but also to differentiate into the many different lineages of LGR5 negative cells. So, so another reason we've been really excited about studying the intestine is because one has many robust assays. So, for example, using this LGR5 reporter mice, one can isolate LGR5 positive stem cells and their Paneth cell niche from mice that are on different, for example, diets, and assay the ability of the ability of these stem cells and Paneth cells from these different diets to give rise to organoids or mini intestines in culture, where one essentially embeds cocultures of these stem and Paneth cells in matrigel, and in the appropriate culture conditions, these stem cells give rise to clonal mini intestines, shown here in schematic. And as shown in schematic, these mini intestinal organoids have a morphology reminiscent of the morphology of the mammalian intestine, where one has these cryptomains and pseudovillus domains, and on top of the schematic is an intestinal organoid derived clonally from an LGR5 positive stem cell, and one can appreciate that these LGR5 GFP positive cells, just as they do in vivo, in these intestinal organoids reside at the bottom of these intestinal crypts. So these mini intestines, these mini intestinal organoids really faithfully recapitulate the cellular diversity that one observed in vivo, because this can be demonstrated using multiple techniques, two of which I'm showing here. By electron microscopy, one is able to morphologically confirm that indeed all of the main intestinal lineages are present within these organoids. Moreover, one can use, for example, single-cell multiplex qPCR and confirm that indeed these mini intestinal organoids possess LGR5 positive, olfactomedia positive intestinal stem cells. They possess chromogranin A positive hormone secreting cells, MUC2 positive goblet cells, and lysozyme positive Paneth cells. And we've been really interested in leveraging this organoid technology, for example, to understand how dietary components, for example, of a high-fat diet alter stem cell function and influence tumorigenicity in this controlled ex vivo setting. So, to gain insight into how obesity might be altering the biology of these intestinal stem cells, we chose to model obesity using a high-fat diet, keeping in mind that this is only one diet which models obesity, and that there are other types of diets one can use, but we wanted to start with the high-fat diet. And when one ages these mice on a high-fat diet, where these mice receive about 60% of their calories from fat, these mice gain around 50 to 60% more mass than their control-fed counterparts. But interestingly, if one ages these mice for more than a year, one finds that these obese mice develop intestinal cancers, shown here grossly and microscopically, consistent with epidemiologic data in humans, where there is this link between obesity and intestinal tumorigenesis. So an important question that arises from this observation is, how does a high-fat diet impact intestinal stem cell biology? So one can address this question and ask, how does a high-fat diet quantitatively impact intestinal stem cell numbers? And in data that I'm not showing you, we've shown that a high-fat diet leads to about a twofold increase in the quantitative numbers of these LGR5 positive cells in the high-fat diet. But another question one can ask is, how does the quality of these stem cells change? And this is really interesting because one can, again, leverage use of these organoid assays to understand how this diet is changing the function of these stem cells. So here we're looking at, on the vertical axis, we're looking at the ability of an LGR5 GFP high stem cell to give rise to an organoid body, and on the horizontal axis, we're looking at stem and pana cells from the different dietary conditions. So consistent with what Hans Cleaver's lab has shown and what we've previously shown, control stem cells by themselves are ineffective at giving rise to these mini intestines. However, when one co-cultures these stem cells with their pana cell niche, the presence of the niche cell dramatically boosts the potential of these stem cells to give rise to these organoid bodies. Interestingly, stem cells derived from high-fat diet mice, these stem cells are now poised to give rise to these mini intestines. And this is really exciting because it suggests that perhaps one of the earliest changes that occurs in response to the high-fat diet in the stem cells is this acquisition of niche-independent growth, or uncoupling of stem cells from their pana cell niche. And not only does one see this in the organoid assay, but histologically in tissue sections, one finds that in the high-fat diet intestine that there are more stem cells and less pana cells. So we think that an early change that's occurring in the intestine in response to this pro-obesity diet is an uncoupling of stem cells from their pana cell niche, histologically as well as functionally in the organoid assay. And lastly, these stem cells still respond to cues that are emanating from their pana cell niche, because these high-fat diet stem cells further increase their ability to they increase their ability to give rise to these mini intestines when co-cultured with their pana cell niche. And here are just representative organoids from control and high-fat diet fed mice. So we were really interested in understanding, do dietary components of the high-fat diet mimic aspects of in vivo high-fat diet feeding? So we asked a very simple question. Just as a high-fat diet leads to more intestinal stem cells and this acquisition of niche-independent growth in the organoid assay, does palmitic acid, which is a main constituent of the high-fat diet, mimic some of these effects? And to address this question, we cultured naive intestinal organoids in the presence of palmitic acid or vehicle for 3 to 4 weeks, where we would serially passage these organoids weekly in the presence of palmitic acid. And by immunofluorescence, one can appreciate that there's more green upon long-term exposure to palmitic acid. And remember, green is a proxy for LGR5. Moreover, one can use flow cytometry and analyze hundreds of organoids, and when one does this, one finds that there's about a twofold increase in the overall frequency of these GFP high stem cells relative to their controls. And importantly, functionally, upon long-term exposure to palmitic acid, these stem cells are more likely to give rise to organoids compared to their vehicle-treated counterparts. So we don't think that these effects are necessarily specific to palmitic acid. We're able to recapitulate these with other dominant fatty acids of the high-fat diet, such as oleic acid. We've used commercial lipid mixtures that contain many of the fatty acids of the high-fat diet, and we think that these effects aren't specific to mice because one is able to recapitulate these using patient-derived or human-derived organoids. So what is the mechanism? So to address this, we performed RNA sequencing on the stem and progenitor cells from high-fat diet and control mice, and a pathway that really jumped out at us is the PPAR pathway, and these PPARs are nuclear receptors that sense increases in endogenous ligands, which are derived from fatty acids, and drive a very robust metabolic fatty acid oxidation program, enabling cells to use lipids as an energy source. So we asked two simple questions. Is this PPAR delta program necessary and sufficient for mimicking the effects of a high-fat diet? So to test for sufficiency, one can administer a very potent activator of PPAR delta, GW501516, and when one administers this agonist to mice for 3 to 4 weeks, one finds that, for example, synthetic activation of PPAR delta in vivo, for example, leads to the stem cells acquiring this ability to give rise to organoids in a niche-independent manner. And here's just representative day 12 organoids from control and GW-treated mice. And in data that I'm not showing you, we also have shown that GW treatment increases the in vivo frequency of these LGR5 positive stem cells. And not only is PPAR delta sufficient, but when one makes PPAR delta knockout organoids, one is able to block the organoid enhancing effects of lipids such as palmitic acid and our commercial lipid mixture, as well as block the effects of our PPAR delta agonist, indicating that PPAR delta mediates fatty acid-driven organoid self-renewal. So I just want to circle back as I finish my portion of the webinar by coming back to the question of how does obesity contribute to the early steps of intestinal tumor incidence. So we've known for the past decade or so that many adult tissues are maintained by adult stem cells, shown here in red, and these adult stem cells often acquire the initial mutations that go on to give rise to cancers. So what's happening in obesity? We think what's happening in response to a high-fat diet in the intestine is one sees expansion of the intestinal stem cells, shown here in red again, and because one has more intestinal stem cells, one has more substrate that can undergo oncogenic transformation. But we also hypothesize that perhaps non-stem cells or non-stem cell progenitor cells in the intestine can undergo oncogenic transformation. And the reason we hypothesize that is for the following data. Again, utilizing the organoid assay. Up until now, I showed you data showing the effects of a high-fat diet on stem cells. But what about on the non-stem cell progenitor cells? Interestingly, here we're looking at the ability of non-stem cell progenitors to give rise to organoids from our different conditions, and one finds that these progenitors from high-fat diet, as well as from GW-treated animals, acquire the ability to give rise to these mini-intestines in culture, showing that this high-fat diet PPAR delta program confers organoid-initiating capacity to non-stem cells. And remembering that this ability to initiate organoids is often something we ascribe to intestinal stem cells. And lastly, an important question is, well, these non-stem cell progenitors can give rise to these mini-intestinal organoids, but can they undergo oncogenic transformation and transfer disease to recipient mice? To do this, we synthetically activated PPAR delta with our agonist and deleted a tumor suppressor gene, APC, which is often mutated early on in intestinal cancers. And we performed an in vivo orthotopic transplantation assay that we've optimized in the lab. So when one looks at the vehicle-treated stem and progenitor cells that are APC deficient, only the APC null stem cells are able to transfer tumors to recipient mice. Interestingly, when one activates PPAR delta and then deletes APC in stem and non-stem cell progenitors, now both stem and progenitor cells can transfer disease. So we think this is really exciting because it indicates that this metabolic program activated, for example, by this high-fat diet has potential therapeutic implications for patients where targeting PPAR delta or its downstream effectors in the appropriate dietary or metabolic context might lead to possible treatment interventions where we think, for example, a fat-rich diet, to summarize, we think that a fat-rich diet or mimetics of a fat-rich diet, in this case, in the intestine, we think our PPAR delta agonist leads to PPAR delta activation, which drives activity of stem cells and progenitor cells, which leads to cancer. And from a therapeutic standpoint, we're really interested in asking whether in vivo inhibition of PPAR delta can block the genesis of tumor formation or perhaps be exploited to treat tumors that arise in obesity. And just to leave you with really the two conclusions that we were able to glean, in part by using organoids, we think that these intestinal stem cells, as a consequence of long-term high-fat diet, gain niche independence, and that non-stem cell populations within the intestine acquire stem-like properties. So with that, I'd just like to thank our many collaborators and individuals in my lab, particularly graduate students Samir Beaz, Mieko Mana, and Jayton Roper, who really generated all of the key data that I showed you. And with that, I'll turn it over to Shri and Marie.
Thank you, Omar. That was very interesting. And thank you to our two speakers for sharing their exciting discoveries. We're going to now take a few minutes to go over some of the questions that our listeners have submitted. We'll direct a few questions to you, Madeline, first, and then next to Omar. So I think the first question is to Madeline. Do you think it will be possible to scale up neural or brain organoids for things like drug screening?
That's a really great question. I think that would hopefully be something in the future. At the moment, I think it's a bit difficult because of the heterogeneity, and even in situations with, for example, the cortical spheroids, I think it's still probably a bit early days. But there has been some recent work trying to scale up production of organoids in general, and that would probably be one of the first steps. So, for example, the group of Guoli Ming and Hong Jun Song, where they've been able to make sort of mini spinning bioreactors and therefore generate much larger numbers of these types of cerebral organoids. So I do think that it may be possible. I think we need to overcome the heterogeneity issues, but I think we're getting there.
Great. Let's move on to the next question. So, Madeline, do you think that tissue engineering scaffolds, particularly solid porous scaffolds, would limit the self-organization ability of neural stem cells to form brain-like structures?
I think that's a really great question. I think that one of the really interesting discoveries and sort of realizations that these various organoid methods have given us is that when you allow the cells to completely self-organize in the sense that they just touch one another and therefore sort of sort out into their own domains, you end up with these tissues that look remarkably similar to the in vivo tissue. And it's kind of a different way of thinking than the typical tissue engineering approach where scaffolds are used as a way of trying to build it kind of like you might build a building with bricks of different types. And so I think that a large kind of scaffold, like brain-shaped scaffold, for example, I think that would probably disrupt the self-organizing capacity. But perhaps some kind of combination of the two in a way that still allows the self-organizing capability would allow for at least some shaping of some sort, and that's something that we are looking into.
Thank you. There's one more question for you, Madeline, which is how could one have myeloid cells, microglia more specifically, to become part of the cerebral organoids in vivo?
Mm-hmm. So as many may be aware, or may not, I don't know, microglia are actually not generated from within the brain. During very early embryonic development, they're generated in extraembryonic yolk sac tissue, blood islands. And so, as you might imagine, we don't really think that we have these cells already in the cerebral organoids because of the fact that we sort of push them away from other germ layers, and we don't really think we get extraembryonic tissues in there. But perhaps with a separate protocol to generate these, I think that there are beginning to be protocols for generating these types of cells in vitro from human and mouse ES cells. One could perform coculture type experiments, and that would be something to look into.
Excellent. So we'll move on to Omar for a few questions. So one of them, what role do you think the gut microbiome might contribute to the high-fat diet organoid phenotype?
Sure. Clearly there's been a lot of work done on how obesity and high-fat diet alters the microbiome, and these changes in the microbiome correlate with a lot of physiologic changes that we attribute to obesity. So we're now working on this question where we have germ-free mice on a high-fat diet, and we're trying to understand how much of this changes that we observe in the gut epithelial, stem, and progenitor cells, how much of this is a direct result of the changes that are occurring in the microbiome. So we're really excited. We think that's an exciting direction, and we've now moved into that direction.
So as a follow-up question, are there attempts, or is it possible to just then co-culture the bacteria with, your organoids?
Yeah, I mean, I think that is possible. It's been shown that there are protocols, for example, co-culturing H. pylori with gastric organoids, and there's some work now trying to grow intestinal organoids in the presence of components of their microbiome. So I think we're in the early innings, but there's a lot of groups are working in that direction.
Great, thanks. So, sort of touching a little bit about the environment, have you looked at, or do you have know of other studies looking at the role of microenvironment in the context of more cancer organoids, or in your system?
Yeah, I mean, the microenvironment is very complex because you've got many different types of immune cells, changes in endothelial cells, changes in myofibroblasts, and in just regular fibroblasts. All of these likely undergo changes in different diet-induced physiologies. So, one approach could be to start to isolate each of those components and test them specifically in the organoid assay. Unfortunately, it's still early to do, those types of studies. Our group and a lot of other groups are working in that direction. But another thing that our group has been working on is we've really spent a lot of time, and I couldn't talk about it today, but using transplanting organoids from different physiologies into mice that are, for example, on a high-fat diet or on a regular diet or on caloric restriction, and transplanting these organoids into that microenvironment and, following it by immunofluorescence and then harvesting those organoids and doing functional studies. So, right now our approaches, we've been really working on this endoscopic transplantation model of organoids to better appreciate and distill out what role the microbiome is playing. But I think ultimately, long-term, it would be nice if one could do those types of experiments in the organoid assay.
Great. So I think what we have here is a technical question, really, which both of you are welcome to answer. The question is, both techniques presented use Matrigel as a component to aid in cell culture, and have either of you used more defined components? And the examples provided are fibronectin, biturinectin, laminin, collagen, or maybe a combination of these. So maybe Madeline, do you want to comment first?
Sure. Well, my simple answer is no, honestly. We haven't tried anything other than Matrigel at the moment. I think that's going to be something that probably will need to be worked out, particularly for future types of therapeutic uses, because Matrigel is animal-derived. It's from a mouse tumor. And so, yeah, I think it would be interesting to try other things and see which aspects of Matrigel might be the important features. It's a combination of laminin, collagen, fibronectin, many different extracellular matrix components, as well as probably lots of unknowns. So I don't know, but maybe Omar has tried other possibilities.
Yeah, we've played around with it, but unsuccessfully. So, we've really we're really right now relying on the use of Matrigel. We've tried different combinations of collagen type 4, fibronectin, hydrogels, but so far we haven't found a robust combination.
So we have another question that I think both of them, there's quite a bit of interest in this as well, but so can you both, either of you, comment a bit more on what the challenges in sort of vascularization of the organoids are, and what the specific issues are and limitations?
So I could start on that, I think. So I think there are many issues here, because with vascularization you're talking about not only perfusing the tissue with some type of tubular structure or something, which you might imagine you could perhaps introduce through microfluidics or something like that, but not only that, I mean, the vasculature in vivo provides a blood supply, so you would need something that would be a good enough sort of blood substitute. And then you would also need something that would pump it in a reliable way that would be mimicking the kind of flow that you would get from the heart. So there's a lot of features there that I think are going to be difficult to overcome, and I think that probably the tissue engineering field has been trying to tackle for much longer than even the organoid field has even been around. So it's one of those kind of things that I hope that one day we will tackle it, but I think we still need some pretty important big discoveries. But that said, I think there are other approaches that people are starting to use which would encompass sort of ectopic transplantation approaches. And we haven't done any of this with brain organoids, but for example, liver buds, for example, that have been generated by a group in Japan where they've, Takanori Takebe, where they've been able to transplant these into various ectopic sites in mice and get vascularization. So I think that's something that could be interesting to look at in the future.
Yeah, I agree with Madeline. I mean, the approach we've been taking now is we're doing orthotopic transplantation using colonoscopy into the endogenous environment, and we're really excited in using that technique. But recapitulating this in a dish has a lot of technical challenges that Madeline touched on.
Great, thank you. So Madeline, I think this is for you. Have you tried making cerebral organoids from other species, and do you see differences?
So that's actually something that's an important focus of my laboratory now that I have my own independent group. We're very interested in looking at particularly evolutionary questions, and we are starting to look into a number of different mammalian species. But actually the group of Rick Livesey here in Cambridge has already started this and published recently work where they've done quite a bit with neural rosettes from a number of primate species, so from human, chimp, and macaque. And they're able to see some important differences there. And they also do produce forebrain organoids according to the Kadoshima protocol that I talked about. So, and they do see differences particularly between the human and the macaque. So I think that's a route that is hopefully going to be fruitful in the future.
And this is a question for Omer, sort of touching on variability. So do you see a lot of differences, variability between the size and differentiation of intestinal organoids within a single culture?
Yeah, I would say, I mean, there definitely is variability, and, this is why, when we, for example, quantitate, stem cell frequency in the organoid assay, our preference is to use a technique like flow cytometry, where one can analyze hundreds of organoids at once. There's also variability in terms of the culture conditions that one uses. For example, in the intestinal system, one uses Wnt and agonists of Wnt, and, based on the concentration of those growth factors that one uses, that can impact the frequency and biology of the stem cells as well as their differentiation. So I think it's important to use consistent culture conditions, and when doing quantification type experiments, we try to use flow cytometry and get averages from hundreds of organoids rather than relying on counting from 10 or 20 organoids manually.
Madeline, there's a question that came up, which is how can we use organoid-based modeling to study developmental disorders, especially neurodevelopmental disorders?
So that's actually something that we did a little bit while I was still in Vienna. We looked at the disorder of microcephaly, and there we were able to take patient iPS cells, in fact, and generate organoids that showed features of microcephaly, particularly that they were much smaller and showed defects in their neurogenesis. Specifically, they showed a premature neural differentiation. That was, I think, the first use of these types of brain organoids for a neurodevelopmental disorder. But since then, now the group of Flora Vaccarino has used forebrain organoids, using the method originally established by the Society group, and they also were able to. They also used patient iPS cells to generate their forebrain tissues, and then look at their development, and they could see an imbalance in the various neural types that were generated. And I think that we are going to see in the future a large number of different disorders that are looked at using this approach, and hopefully it will prove valuable in those studies. Most recently, there have been quite a surge of studies using brain organoids to look at the effect of Zika virus. And there again, it's been able to give us some insight, particularly showing that the neural stem cells seem to be very sensitive to Zika virus.
And do you think that we're still quite far away from modeling diseases where there aren't any gross morphological phenotypes?
Well, I think that, of course, if you go later in development or in the lifespan to disorders like schizophrenia or even later like neurodegenerative disorders or things like that, they may prove more difficult. But even schizophrenia, for example, has a neurodevelopmental component. And so at least that aspect probably could be studied. And there are also approaches, I think, to potentially use in vitro methods to look at even neurodegeneration, where you might be able to kind of artificially push the neurons to age in a way that allows you to model these types of neurodegenerative disorders. So I hope that in the future we can also use them to look at more late onset disorders, but we'll have to see.
Great, thanks. And for our last question, we'll switch over to Omer. So how do other lipid components of the high-fat diet influence stem cell and organoid growth?
Yeah, so we're actively looking at this. It's a complex question because, there's tens of hundreds of different lipids, and, we're really interested in figuring that out. Right now, we only have concrete data for some of the main components of the high-fat diet, but we're also looking at lipids that aren't found in the high-fat diet. And, so right now we're still in discovery mode. But we are leveraging this organoid assay to address exactly those types of questions because, we want to understand which of these lipids are instructive and important regulators of intestinal stemness, and which lipids are just permissive and act as, for example, an energy source.
Great, thanks. And with that, we've come to the end of our session. We'd like to say thanks again to our speakers, Madeline Lancaster and Omer Yilmaz, for their engaging presentations. We're very grateful to our sponsor, Biotech, for the contributions that made this webinar possible. We'd also like to thank you, the audience, for tuning in and asking great questions. If you missed anything during this webinar, or if you would like to listen to it again, a recording will be available shortly on the Cell Press website. If you have any comments about the webinar or suggestions for future topics, we'd love to hear from you. You can email us at cpwebinars@cell.com. So keep your eyes open for our future webinars and also the Cell Symposia in 2016, which include the 10 years of iPSCs, and also, of course, for our Organoids lab links in Amsterdam. Thanks a lot, and goodbye.
Bye.